The root has a radial structure. It possesses radial symmetry due to the presence of radial vascular bundles. The root can be divided into zones that differ in function. The zones of cell divisions, zone of elongation, and zone of maturation. The transitions between them are gradual without distinct boundaries. At the tip of the root lies the zone of cell division, which is about 1 mm long. It consists of meristematic tissue, the apical meristem, protected from damage by a thimble-like root cap. New cells for the future root tissues are formed here. The cells of the root cap live for 4 to 9 days and, as they break down, are replaced by the new ones. The root tip, along with the cap, pushes between soil particles. To facilitate sliding, the cap is covered with mucus. In the zone of cell division, the root skin, as well as the primary cortex at the central cylinder, begin to develop. The tips of adventitious roots in floating aquatic plants do not have a cap but are instead covered by root pockets that protect cells from unfavorable conditions and small aquatic animals. Above the zone of cell division is a small zone of elongation. Its length is only a few millimeters. This is where cell growth occurs, causing the root to lengthen. Consequently, the root tip, which includes the zones of cell division and growth, is constantly advancing through the soil. Next is the zone of maturation, which is up to several centimeters long. It is characterized by an abundance of root hairs and is sometimes called the root hair zone. These are tubular outgrowths of the outer skin cells. Their length ranges from 0.15 to 8 mm, and their lifespan is 10 to 20 days. The significance of root hairs is immense. They enable plants to absorb water and dissolved nutrients. Thanks to these hairs, the absorption surface area of the root increases. They also perform a mechanical role, serving as support for the root tip. There are a vast number of these hairs on the roots of every plant. Root hairs are short-lived. As the root grows, they quickly form at the bottom of the absorption zone and die off at the top. Higher still is the zone of conduction, for the zone of lateral roots. Lateral roots form as branches of the primary root, and they can further branch out, forming a complex root system. Lateral roots originate in the internal parts of the parent root, within the pericycle, which is why they are called endogenous. In angiosperms, cells of the pericycle and endodermis participate in the formation of a lateral root. Gradually, the lateral root increases in size, passes through the primary cortex, penetrates the soil, and immediately begins to form a root cap, as well as apical and primary meristems. Lateral roots do not form simultaneously. They usually initially develop from areas of the pericycle located opposite the vascular bundles of the primary xylem and therefore arranged in regular rows along the parent root. Later, lateral roots form from other parts of the pericycle and their regular arrangement is disrupted. Above the conduction zone is the root neck, which connects the root to the stem.